Hi everyone, welcome back to your online lecture. This chapter, chapter 10, is entirely on photosynthesis. Photosynthesis is, as we're going to see, a very special process because it is able to transform solar energy from the sun into something physical, organic molecules, which we are able to use to make energy from. Photosynthesis occurs in special organelles called chloroplasts. If you haven't yet gone over cellular respiration, be sure to go check out that lecture because there are a lot of similarities between cellular respiration and photosynthesis. We're going to be going into the molecular aspects of it, and if you understand cellular respiration, it'll just make photosynthesis that much simpler. So for this chapter, the objectives are that students will be able to explain the processes of energy transformation relating to photosynthesis, particularly be able to describe the ecological relationship between autotrophs and heterotrophs, describing fundamentals of endosymbiotic theory, and model or map the events occurring in photosynthesis. So, as I mentioned, this entire chapter is focused on the process of photosynthesis. And why do we have an entire chapter focused on photosynthesis? It's because of how special this process is. It is, as defined here, the process that converts solar energy from the sun into chemical energy. And with chemical energy, what we mean is that it makes certain organic molecules, right? We can then take that chemical energy, those molecules, and utilize them in our own processes to make energy, right? Let's look back at an energy diagram from the previous chapter. This is from chapter 9 on cellular respiration. So here we have a cycle, and on the top we have the reaction for photosynthesis, and on the bottom we have the reaction for cellular respiration. So let's look at photosynthesis. We know that plants need water and they need access to good air, and in that air there is carbon dioxide. So plants take the carbon dioxide and the water coupled with light energy from the sun, and they're able to take this light energy, and the chloroplasts are converting it into organic molecules and oxygen gas. And organic, remember, means anything having to do, it's a carbon-based molecule. So the organic molecules we're talking about here are the macromolecules from chapter 5, carbohydrates, lipids, proteins, nucleic acids. So plants are able to steal energy from the sun and turn them into something physical, a molecule that we can use here on Earth. So then we have organisms that contain mitochondria, such as animals. Plants also do. Just we'll see that again, but we'll use us as an example. We are cellular respirators. We have mitochondria in our cells. So we eat, we take those molecules and we break them down. We oxidize them, and along with oxygen gas, and the mitochondria are able to turn those into ATP, which is adenosine triphosphate. That is our energy molecule. And what they expel is carbon dioxide, right? This is what we exhale, and water. Water is a byproduct of a lot of our biochemical reactions in our body. But what you really need to see here, which is, again, so special about photosynthesis, is that the input and output here, the input of energy is, again, light energy from the sun. They use photons, and they can turn that into a physical molecule, matter. And then the output here is, in the process of making ATP, only about 30% of the energy is utilized in making ATP. About 60% is lost as heat, which you can feel, right, when you go near an organism. So because of this ability to take energy from the sun, photosynthesizers are said to feed the biosphere. Directly or indirectly, photosynthesis is nourishing almost the entire living world. So what are some examples of organisms that are able to conduct photosynthesis? Obviously plants, right? There are some photosynthetic algae. There's a couple of unicellular eukaryotes. So all of these have organelles. And then prokaryotes, remember, do not have organelles. So the organelle responsible for photosynthesis is the chloroplast. So plants and eukaryotes will all have chloroplasts. Prokaryotes do not have chloroplasts. So they will have some of the same machinery, what I mean by that is proteins that are able to do all of those biochemical reactions. They just won't be contained within the chloroplast. So just keep that in mind. But all of these are able to conduct photosynthesis. In this schematic, you'll notice that there are two types of organisms, right? Two basic types. We have one which is able to create food or energy, and we have one that can't create its own. It needs to take it from another organism. So there are specific terms for these, and that's autotroph and heterotroph. So an autotroph, auto means self-feeder. Autotrophs sustain themselves without eating anything derived from another organism. So in this example, autotrophs are those that are able to photosynthesize. Another way that we refer to autotrophs is saying that they are the producers of the biosphere, because they're the ones creating those organic molecules from carbon dioxide and water, right? Almost all plants are autotrophs. They're a specific type of autotroph called a photoautotroph. Photo because obviously they use photosynthesis uses solar energy to make their own organic molecules, but an example of a plant that is not an autotroph would be something like a Venus flytrap, which is able to capture flies, right? And flies are other organisms. So this would be an example of a plant acting as a heterotroph. So this is the other type. Heterotrophs obtain organic material from other organisms. So while the autotrophs are the producers, the heterotrophs are the consumers of the biosphere. So they eat other living organisms in order to survive. And there's a special subset of heterotrophs called decomposers, which specifically eat dead organic material or feces. So maybe like a vulture or some forms of mushrooms. But again, since heterotrophs are the consumers, they depend on autotrophs. For example, photoautotrophs, plants, for food and oxygen. In the event of something catastrophic happening which would block our access to the sun, you might not think this is a big deal, all right? We don't need sunlight to survive. I mean, there's some things that we need, vitamin D, but what it would do is it would stop any plants. Just think of any food that you eat, any vegetable, and then obviously the livestock that we eat also rely on plants. So we would eventually run out of food because our producers, our autotrophs, wouldn't have any more of their solar energy to utilize to make chemical energy. So let's get into the process of photosynthesis. Here we have the organelle responsible, the chloroplast. Note that the chloroplast is a double-membraned organelle. There is an inner membrane and an outer membrane, just like the mitochondria. So photosynthesis tends to happen in the leaves. The leaves are the major location for photosynthesis in plants. And chloroplasts are found mainly in the cells of the mesophyll, which you see here, which is the interior tissue of the leaf. Each mesophyll contains anywhere from 30 to 40 chloroplasts. And the carbon dioxide, which the cell needs as a reactant in their process, the carbon dioxide enters into a pore called a stomata. And then the oxygen that the plant creates as a byproduct also is able to leave out of that very same pore. And again, that's the stomata. So you see carbon dioxide entering and oxygen leaving. So within the chloroplast, within the membranes, there is a dense fluid that fills it called the stroma, right? So that's what you see there. And you'll also see that there are these stacks of green sacs, and those are called thylakoids. Thylakoids are stacked into columns, which are called granum. So each of these, they look like little cookies to me. Each of these little chips or cookies that are stacked is the thylakoid, and then a whole stack of them is called the granum. And what makes plants green is a pigment called chlorophyll. And that's a really important part of the process of photosynthesis. And there are actually a couple types of chlorophyll, but just note that chlorophyll is the pigment that gives the leaves their green color. It resides in the thylakoid membrane, so that's why they're green, and it's very important for the process of photosynthesis. In the last chapter, we went over the importance of redox reactions, reduction and oxidation reactions, because they are what drive the part of cellular respiration called oxidative phosphorylation. Well, and it all has to do with the movement of electrons, right? That's really what we're doing with any kind of chemical reaction. And the same thing is happening here. So you had to learn the reaction for cellular respiration in the last chapter, and in this chapter you have to learn the reaction for photosynthesis, which is actually almost the reverse of cellular respiration. So you have 6 CO2, 6 carbon dioxide molecules, plus 12 H2O, plus the light energy from the sun, yields C6H12O6, which is our organic molecule. This is some type of sugar, for example, glucose, plus 6 O2, which is oxygen, plus 6 waters. Now, it is a little bit different from the cellular respiration one. If you were to, what they figured out a little bit later on that photosynthesis just requires a couple extra waters to get it going. But it also creates waters. So if you were to balance out this equation, you could cancel out 6 waters from here, 6 waters from here, so this will be gone, and this will now read 6 H2O, and then that will be the exact opposite of cellular respiration. So it's very similar, it's just that photosynthesis requires a couple additional water molecules to get it going, and then it produces, it kind of kicks them out. It uses them and then creates some. So it does neutralize and balance out. So just note, right, the reactants are carbon dioxide and water, and what we're producing is as a glucose or C6H12O6 and water and oxygen. As a quick refresher on redox reactions, this has to do with the movement of an electron. We have two essentially opposite reactions. We have something that gives away an electron and something that accepts an electron. So when we lose an electron, that's called oxidation, and when we gain an electron, that's called reduction. So together, this is a redox, reduction, oxidation, a redox reaction, because if one is happening, the other also has to happen, right? If I'm holding an electron and I give it to somebody else, I was oxidized, I lost my electron, and they simultaneously were reduced. So they occur simultaneously. So this transfer of electrons is what happens during chemical reactions, and it is what releases the stored energy in organic molecules. So here's a very basic example. You have an X that's holding an electron and a Y. Through this reaction, the X loses an electron, so it becomes oxidized, and the Y is going to be what's accepting or gaining this electron, so it becomes reduced. So you have to be aware of what is being oxidized and what is being reduced in the reactions that we're talking about. I went over the cellular respiration one in the last lecture, so look that over if you're still a little bit confused. And here is the reaction for photosynthesis. So we have our light energy, carbon dioxide and water. So carbon dioxide is becoming reduced. It is gaining electrons. And when we gain electrons, remember, electrons are negatively charged. We don't want to just stick negatively charged things on our molecules, because that creates a new molecule that is reactive. And if you just have things with electrons hanging around your cells, they become oxidants, right? And that's why we eat antioxidants. We want to get rid of oxidants, things that are going to oxidize, things that are going to rip away other cells, other parts of the cell. So we don't just want to stick electrons on here and have them negatively charged. So with electrons, we tend to have other things that will attach onto here, particularly often hydrogens. So if you're wondering how that happens, because hydrogens are just a proton, so we'll just have a proton and the electron, because protons are positively charged and electrons are negatively charged, so they do neutralize each other. So the carbon dioxide gas is going to gain some electrons and become reduced, and also grab some protons in the process, and we built a larger molecule. On the other hand, the water molecule is becoming oxidized. It's going to lose its electrons. So it loses electrons, and it also loses some protons, some hydrogens, and becomes O2. And we are going to actually see how important the splitting of water is for the process of photosynthesis. But do note and be able to answer the question of what happens to water during photosynthesis. Is it becoming oxidized or is it becoming reduced? What about the organic molecule? Was that reduced? Does it become oxidized? So this would be the cellular reaction for that. As far as all of those redox reactions, photosynthesis is reversing the direction of electron flow compared to respiration, right? So respiration's going this way, photosynthesis is going this way. So we say that that's reversing the direction of electron flow. It's a redox process in which water is going to be oxidized. We're going to rip the electrons right off of water, and carbon dioxide is being reduced. We are building up carbon dioxide into a much larger organic molecule. And this is an endergonic process. Remember from chapter 8, we had exergonic and endergonic. And exergonic reaction is one in which energy is released from the reaction, and then an endergonic reaction is one in which energy is required for that reaction to occur. So photosynthesis is an endergonic reaction. We need to give it some energy in order for that to occur, and that energy is provided by sunlight. So just like cellular respiration, photosynthesis has a couple of stages that we're going to go through. Particularly, it has two stages: a light reactions stage and a dark reactions, as we call it, but it has a specific name called the Calvin cycle. So Again, just like with the cellular respiration process, there are a couple of important things that you need to pay attention to when we go over these. One will be the location. Where is this occurring? Two will be the reactants. So what is going into the process, and the products are also important. We're going to have an electron transport chain, just like in cellular respiration. So make a table. That's the best that I would say for this, a table to help manage all of this information as we go through it, and I'm going to call it out as we go. So here is a schematic of the light reactions and the dark reactions. And I also just want to note that that doesn't mean that they occur at night versus during the day. These are called the light reactions because they're the part that need light. So these can only occur during the day. But the dark reactions, they don't occur at night. They just don't require light. So they could happen during the day or during the night. But the light reactions do need sunlight. So here is our chloroplast. Remember, it's got the double membrane. We have the intermembrane space between the two membranes, and then we have the inner part, which is the stroma. Within the stroma, we have the thylakoids. Each one of these is a thylakoid, and a stack of thylakoids is a granum. So this is a granum. So the light reactions are going to be occurring in the thylakoids. And remember, they're green because they contain chlorophyll. So we're going to be using light here. This is where water is going to come into the reaction. And we're going to kick out oxygen because we are breaking down. We're going to oxidize water. So it's going to lose some electrons and lose some protons. The electrons and protons stay over here for the reactions. The oxygen gas is released out of the stomata, right? Well, the stomata is part of the leaf to, exit into the atmosphere. So that's the light reactions, and we're going to go into the molecular parts of this, but just as an overview. The light reactions used these products, NADP+, which is a new molecule that we'll see, and then ADP and inorganic phosphate, right? This is what we pop together to make ATP. So we fed those in, and ATP is made here. We feed this in, and it's going to grab some electrons. It's another electron transporter, just like NAD+. It's just that this one is specific for plants and anabolic reactions. And then the second phase is the dark reactions, or the Calvin cycle. So what we see feeding into the Calvin cycle, we have carbon dioxide gas. So this is where this comes in. It goes through this cycle. We use ATP, and the electrons that were gathered by this electron shuttle, they get fed into this cycle, and the carbon dioxide gets reduced. It's going to grab all of those electrons. It's going to be built up larger, and we are going to create a larger organic molecule. For example, C6H12O6, which is a type of carbohydrate, which is a sugar. And then because we use the ATP, we're going to kick out our products, which we broke it apart. So it's an inorganic phosphate and an ADP. We are using the NADPH, which is carrying electrons. We use them, and then we kick out the NADP+. So in this way, the ATP and NADPH is in a reusable, renewable cycle here, which is good. And then we have our reactants for photosynthesis up here, and we have our products for photosynthesis down here. So that's the big, snapshot of this. We are going to go into the actual reactions of how this occurs now. But first, we have to go over of the most important part of this, which is light. We're going to look a little bit into the nature of sunlight. So we know that the light reactions convert solar energy from the sun into chemical energy. It's helping make molecules for us, and the molecules that it makes particularly are ATP and NADPH, right? Here's the light reactions. They use solar energy. We're making ATP and NADPH. Their thylakoids transform light energy into the chemical energy ATP and NADPH. So let's look at sunlight. Light is a type of energy called electromagnetic. We know that electromagnetic energy travels in these rhythmic waves, and we are able to measure the wavelengths, which is the distance between different parts of the waves, to measure what type of electromagnetic energy this is. We can measure between the troughs or the crests. Here we have a little picture for that. So the crest is the top of the wavelength and the trough is the bottom of the wavelength. They would be the same, right? This distance versus this distance. So you can measure wavelength at either spot. The wavelength will differ based on how frequent this wave is occurring, right? We can have more frequent ones or we can have less frequent ones. And once we measure the wavelength, we place them onto a spectrum called the electromagnetic spectrum. Here's an electromagnetic spectrum for you. So you know that we have some light that is visible, which we can perceive, and then we have some that is not visible. So let's look at this across here. So this is written in scientific notation. So we have 10 to the third meters. So that's 10 times 10 times 10. So that's 1,000 meters is the wavelength, right? So that just the distance between there would be 1,000 meters. That's a lot. So this is the larger, longer wavelengths. And as we move this way, they're becoming smaller and smaller and smaller, which means that the frequency is increasing. So as the wavelength, which is larger here, as the wavelength decreases, the frequency increases. They are indirectly related. Just looking at some of these types of waves, the radio waves obviously are for radio transmissions. Microwaves are in your microwave. What they do is they rotate water molecules. So they create kinetic energy, right? Type of thermal energy to heat up your food. Infrared, IR, if you've ever gone to a spa with infrared. Next we have the visible light, and it goes red, orange, yellow, green, blue, indigo, violet. Do note the wavelengths for visible light. So it's 380 nanometers to 750 nanometers. So I always just kind of think 400 to 750, just as an easy remembrance, but it's 380 to 750 nanometers. And again, these have the longer wavelength. We are getting shorter and shorter and shorter, which means that the frequency, how often they're occurring, is increasing this way. After violet, we have UV, which is ultraviolet, and you know that you can't see that, but it can damage your cells, your DNA. X-ray, again, you can't see that, but it can damage your DNA. And then gamma rays are extremely small. I remember reading a story about gamma rays being present in outer space. The astronauts that were in a shuttle would see little specks of light kind of shooting across their eyes, and they were finding that there were little holes in their suits and in the shuttle, and they were saying that gamma ray radiation was able to break through our barriers. So this definitely would be not good for you either. The really cool thing about light is that it behaves in both a wave and a particle, which is very strange for something to behave as a wave and then something to behave as a particle, which is matter. So light is both both non-matter and matter. It's both wave and particle. And those particles we call photons. A photon is a little packet of light. So this is what we're going to see. The chloroplasts are able to absorb photons for their energy to help push photosynthesis forward. So since we're on the topic of light, we have to also revisit those pigments that I was talking about, because the pigments are what allow for the absorption of light. So here's a chloroplast with its thylakoids and the stack of thylakoids, the granum. Here's light coming through. The pigments that are present in here are able to absorb that light. And we have different pigments which are able to absorb different wavelengths of light. Anything that's absorbed obviously is absorbed, and they use that energy. If it's not absorbed, that energy is either transmitted right through it or it is bounced off and reflected off. So, because we see the color green, that means that that wavelength, which is between 500 to 550, this wavelength of light is obviously not absorbed by these thylakoids. Otherwise, it would be in here. It would be being used for photosynthesis. So thylakoids, the chlorophyll that are in there, are able to absorb all of the other wavelengths of light other than green. That is why we see green. So here are a couple of the pigments that are found in chloroplasts. So again, pigments are substances that absorb visible light. We have photosynthetic pigments. We also have pigments in our body. We have blood that is obviously a red. The heme groups have a red pigment to them. So pigments are substances that absorb visible light. Different pigments are able to absorb the different wavelengths. The three pigments in chloroplasts are chlorophyll A, which is the most important light capturing pigment, chlorophyll B, which is an accessory pigment, and then we have another class called carotenoids. An example of a carotenoid would be beta carotene, if you've ever heard of that, which tends to look orange, so it absorbs all the other ones other than all the other colors other than orange. So carrots and yellow and orange vegetables and fruits have a lot of carotenoids, beta carotene for example. And if you eat too many of these, your skin will get an orangey tinge to it. So here is a picture of chlorophyll. So if you were to look at this structure, this is chlorophyll, and you look at a heme group from hemoglobin in your blood, they look very, very similar. They look almost the same, which is interesting, right? So we have this, which is present in chloroplast, and then the hemoglobin group, which is in our red blood cells. You just see the unity in organisms when you look at the structures like that. So these are called porphyrin rings. This has a magnesium in the center. So this is the pigment that is capturing the photon of light, which the plant is going to use to make chemical energy from that solar energy. It's also talking about accessory pigments such as the carotenoids. What they do is they broaden the spectrum of what this plant is able to absorb. This isn't able to absorb green, so instead the carotenoids might be able to absorb some of that excess light energy so they can use that. And it says it also absorbs excess light energy, which would damage the chlorophyll or react badly with oxygen. So just note, the photosynthetic pigments, particularly chlorophyll A, chlorophyll B, have this porphyrin ring structure. Okay, now we're actually going to get into the processes, the steps of photosynthesis. So we're going to begin with the light reactions, the first parts. First, we're going to cover a couple of terms, specifically what a photosystem is, because this is the complex that's actually doing the work for the light reactions. So this is a photo of a photosystem. So a photosystem is comprised of a couple of things. So in the center, we have what's called a reaction center complex. And the reaction center complex's job is well, it's comprised of an association of proteins in here, and part of that is that it has this special pair of chlorophyll A molecules. So our photosynthetic pigment here, chlorophyll A, and it has something called the primary electron acceptor. So this whole thing is called the reaction center complex. Surrounding the reaction center complex, all this purple stuff over here, are the light harvesting complexes. And they are comprised of a bunch of photosynthetic molecules. They're a bunch of these pigment molecules that are bound to proteins here. And their job is to capture the light energy from the sun, capture these photons, and transfer that energy over to the chlorophyll A molecule here. So that's the light harvesting complex, and it makes sense, right? Because we're harvesting the light photons, that energy from the sun, to transfer to these are our photosynthetic molecules over here, the chlorophyll. You'll also notice that it is part of the thylakoid membrane here. So just like in cellular respiration, our complexes 1, 2, 3, and 4 were part of a membrane. So this is a protein complex, and it's being held in the thylakoid membrane. So all of these green thylakoids, these are filled. These are obviously phospholipid bilayers, right? That are surrounding these, and they're filled with the photosystem proteins. So that's where they all are. I always tell you to pay attention to where things are. So here out on the upper part is the stroma. So this is outside the thylakoid, and in here is the thylakoid space. So this is the interior, the inside of the thylakoid, and position and orientation matters with all of this. So for these light reactions, we actually have 2 of these photosystems as part of our electron flow, and they are called Photosystem 2 or PS2 and Photosystem 1 or PS1. And yes, those are numbered backwards, so remember that. Photosystem 2 comes first, followed by Photosystem 1. And you might say, why did they do that? Well, they found Photosystem 1 first, because this is closer. This is, the end of this part. So if they were backtracking, or they found this part of it first, so they called it Photosystem 1, and then they found the other part, and they called it Photosystem 2. So let's get into the steps for how these photosystems work. Remember, the whole thing about these catabolic reactions is that we are following electron flow. So we're going to see that there are 2 types of flows that are possible within the plant, and it is a linear flow, and we also are going to see a cyclic flow. So the linear electron flow, which is the primary pathway, involves both of the photosystems, and we're going to see that it produces ATP and NADPH using light energy. There are 8 steps in linear electron flow. We're going to walk through each one of these. So step 1, a photon hits a pigment in a light harvesting complex of PS2, and its energy is passed along among pigment molecules until it excites P680. So step 1, a photon strikes a pigment molecule, and it is passed around within the light harvesting complex until it is passed along to this chlorophyll A, and this one has a specific name. It's called P680 because 680 is the wavelength, 680 nanometers. that this chlorophyll is absorbing. So PS2, this photosystem, is associated with the chlorophyll A P680. So that's step one. Photon hits this pigment, the energy, it's energy, right? It's just a ball of light, and it gets bounced along like a hot potato. That's how I always say it. Between all of these pigments within the light harvesting complex until it settles upon this chlorophyll A photosynthetic pigment here. Step 2, an excited electron from P680 is transferred to the primary electron receptor, which now we call it P680+. So that ball of energy, that photon, it's energy, right? So what it does is it excites an electron, and that electron absorbs that energy, and now we have an excited electron. That excited electron gets bumped up to the primary electron acceptor, right? And so now our excited our excited electron is hanging out in the primary electron receptor, acceptor. Step 3 is water is split by enzymes, and the electrons are transferred from the hydrogen atoms to P680+, thus reducing it to P680. So we have just excited an electron, and that electron got kicked up here. That leaves P680 without an electron. So because it lost a charge and a negative charge, it became P680+. It gained a positive charge by losing its electron. So we need to restore that. If we don't, then this won't be able to happen ever again, because there won't be any electron for this to excite. So where do the electrons come from, which is going to replenish this chlorophyll A? This is where water comes in. This is it right here. This is why we need to water our plants. Water is split. This is the water splitting process I talked about earlier. And the electrons get moved onto this P680, which reduces it, right? Reducing is gaining electrons. So water is oxidized and P680 is reduced. That is a redox reaction. Then, because the electrons get kicked off, the hydrogens also leave, and we're left with water. I'm sorry, we're left with oxygen. So this, it says half of an O2, because half of an O2 is just O. So we need 2 of these to happen to create an oxygen gas molecule. And then they associate together, and they're going to diffuse out of the plant through the stomata, and then we can breathe in that oxygen. So you will definitely see a question about how, what is the purpose of water in photosynthesis. So it is the electron donator during PS2 for PS2 photosystem 2 of the light reactions. Okay, and then it's giving its electrons to P680 to replenish what we lost. So we've replenished it. Well, we absorbed our photon. It excited an electron. The electron got kicked up here and it's waiting here. Water came in, was split, and the electrons restored this molecule. Okay, so the next step, step 4, each electron falls down an electron transport chain from the primary electron acceptor of PS2 to PS1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. Does this sound familiar? I hope it does. I hope it sounds familiar. If it doesn't, go back and look at the cellular respiration. Go look at videos on electron transport chain, because this is an electron transport chain. It's just the one for photosynthesis. So we have this excited electron here, and we have a series of cytochromes, which are proteins that are able to transfer electrons for the transfer of energy here. So we have a couple of cytochrome proteins that are going to be passing the electron down. They're going down its energy gradient. We have a high energy here. It's going down its energy gradient. And remember, each one of these is more and more electronegative, which is pulling the electron down to it. As we do this complex, this is creating a proton motive force. We're creating a proton gradient here, just like in cellular respiration. And where does the electron, this is so down the electron chain, where does it fall? It falls onto another chlorophyll A. This one is called P700 because this absorbs wavelengths of 700. So we have two different types of chlorophyll A. So note that chlorophyll P680 is with photosystem 2, P700 is with photosystem 1. So now that this electron is here, we're going to do the whole thing, the same thing all over again. So step 5, I guess we can back up for a second. So that potential energy that's created from that proton gradient, which we're creating, is going to drive the production of ATP by chemiosmosis. So we're creating this proton gradient by this electron transport chain, again, just like cellular respiration. Somewhere along, you'll see a different picture. Somewhere else, there is a ATP synthase enzyme, same exact one as we used for cellular respiration. That proton motive force is driving the production of ATP. So that's where ATP is made, right here in linear electron flow. So that was step 5. Step 6, in photosystem 1, just like photosystem 2, transferred light energy excites P700, which loses an electron to the primary electron acceptor, and then P700 plus, because P700 is missing an electron, accepts an electron passed down from PS2 via electron transport chain. So we've gone through electron transport chain. This has accepted the electron. Now we need to excite this again to kick that electron up. So here's the photosystem 1 absorbing light. That photon is being bounced around between pigments and it gets absorbed by P700. This excites an electron. The electron gets kicked up to the primary electron acceptor. That leaves this P700 plus because it's lost a negative electron charge. Now here we're not splitting water. You know, here we had to split water to replenish it. Here we are not. The thing that replenishes it is the next electron that comes through this chain, right? So the only place we're replenishing with water splitting is in the beginning. And then just the continuous flow of this is going to be replenishing this. So the photon struck, excited the electron, kicked it up to the primary electron acceptor, and once again we have the electron that gets passed to a protein called ferrodoxin, and then finally ferrodoxin passes it to NADP plus reductase. So reductase is an enzyme that reduces, so reducing means to gain electrons, so this enzyme is going to allow NADP plus to be reduced. So in comes our new molecule, NADP plus, and all you have to know is that it's just like NAD, it's just a little bit different, but it's going to function in the same way. NADP plus is often used for anabolic pathways, for building things, and what are plants doing? They're making energy so that they can build organic molecules. So we have NADP plus. It gains 2 electrons via NADP plus reductase and a proton here as well, and we create NADPH. This is an electron carrier which is carrying 2 electrons, and we'll see later on where that goes, so because we're going to make more organic molecules. So that was linear flow, right? Linear is in a line. We have a beginning. Here's the linear aspect of it. It's in a straight line from beginning to end. But I mentioned that there can also be something called cyclic electron flow, where there's a cycle involved. So With cyclic electron flow, here's a nice little picture here. What happens is the electron that's being passed from the cytochrome complex to the P700 chlorophyll A, that gets excited up to the primary acceptor, and then instead of it being passed to ferrodoxin and then NADP plus reductase, ferrodoxin, once it grabs it, moves it back to the cytochrome complex and feeds it back into this electron transport chain. So we can continue, this is a little closed cycle here. We can continue to do this, and in doing so, remember this is pumping protons into a space and creating a proton motive force, which then drives ATP synthesis via chemiosmosis. So by creating this cyclical flow where you can continuously make ATP, we're not making any NADPH, so we can't really bring this over to the Calvin cycle to make sugars, but we can make energy if the plant needs to make more energy. So that is cyclic electron flow. So it only uses photosystem 1 and produces ATP, but not NADPH. There is no oxygen released, right? Because oxygen's released over here at photosystem 2. Some organisms, such as purple sulfur bacteria, have PS1 but not PS2. So this would be how they're producing some energy. Not all organisms have both of these. And cyclic electron flow is thought to have evolved before linear electron flow, and it may protect cells from light-induced damage. So that's just a little adaptation there. And be sure that you know the difference between linear versus cyclic, particularly with what's created, right? And what's used and what's not used. That's a good one to know. Before I get into the dark cycle, the Calvin cycle, we're going to go back over what we just did and look at another version of it. And we're going to compare it to what happens during cellular respiration in the mitochondria, because like I keep saying, they're very similar, and you very well may see questions that are comparing the two. So it's good to see and understand how they're similar. So first, here is a nice picture of photosynthesis. So this is the this is the membrane for the thylakoid, right? So this is the green thing. And out here is the stroma. So as another picture just to show you. So this is the thylakoid. So all of those proteins are in this membrane, and all the lighter color out here is the stroma. And again, it's really important to know what happens where. So the photosystems, photosystem 2 and photosystem 1, are present in the membrane of the thylakoid. So we have light hitting the pigments here. Here's our chlorophyll A. Here's water. Water is coming in at the thylakoid space in here and being oxidized. Oxygen is also released in here, and it's going to just diffuse out. But because water is in the inner thylakoid space here, which is where it's broken apart, the electrons are brought here, the oxygen's going to leave, but the hydrogens are also stuck in here. And remember, hydrogens are what makes something acidic. So this has a very low acidity, a high acidity, a low pH. So it's very acidic conditions because we keep having this replenishment of protons in here. In addition, here's the electron transport chain being taken through one of these cytochromes. Here's the cytochrome complex and another cytochrome over to photosystem 1. So this is the electron transport chain right here. Again, it pumps protons as it's doing that. It's pumping them from the stroma in through to the thylakoid space. So yet another avenue for increasing protons in here. So now this is why this is so acidic. This is the proton gradient, the proton motive force. Remember in the picture we saw that ATP was made. Here's the picture. This is what we're looking at. ATP is made, but it's not made from these proteins. It's made via the proton motive force that these proteins are creating. So now, over here somewhere else, we have the enzyme ATP synthase. ATP synthase is able to couple the proton motive force, the electron transport chain, to the ATP via the proton motive force. So the protons flow through here. This is like a turbine. It's a turbine, it spins, and it creates ATP this way. So, again, knowing where things happen is important. ATP is created in the stroma. So note that. There's no ATP being made in here. The proton motive force is in here. The ATP is in the stroma. And then, photosystem 1, we have ferredoxin and the protein enzyme NADP plus reductase, which is taking NADP plus, taking these electrons which we've just trafficked through here, adding them to this shuttle to create NADPH. And again, NADPH and ATP are both created in the stroma. This is important because the Calvin cycle also takes place in the stroma. So it's good that both ATP and NADPH are being created out here, because then we don't have to shuttle them anywhere. And shuttling them places takes energy, and you don't want to have to make energy and then use energy to move that energy, right? So these are just created out here in the stroma, and then they're present for this cycle to occur. So again, knowing where things take place is important. So that should look very similar to what we saw in the mitochondria for cellular respiration. This is a picture comparing the two. So let's say on the left we have a mitochondrial structure, and on the right we have the chloroplast. So both of them had a membrane which contained the proteins that are part of the electron transport chain, right? So this is their general electron transport chain. Up here was where the proton motive force was being created. In the mitochondria, that was the intermembrane space. So that was between the inner membrane and the outer membrane, remember? And for the thylakoid, for the chloroplast, this was the thylakoid space. That's in here, right? The thylakoid space. So that's the where the proton motive force was created for mitochondria and chloroplast, respectively. The ATP synthase protein was again also in this membrane, and the proton motive force was falling down that enzyme, and the ATP was created on the other side for both of them. So for the mitochondria, this was the mitochondrial matrix. So the most inner part of the mitochondria is where the ATP was being created, whereas for the chloroplast, it was being created in the stroma, so outside of the inner part. So they're kind of opposite as to where things are occurring. But you should be able to understand this, and you'll probably see questions relating to it, just so that you your teacher, if I'm your teacher, if you're in my class, knows that you know where things are occurring, and you can compare similarities across different structures. So this is a really nice picture. Make sure that you can understand this picture. Look at a mitochondria, use this picture and look at the cellular respiration video. That should help you. So let's just see what this says here. Chloroplasts and mitochondria generate ATP by chemiosmosis, but use different sources of energy. So this is chemiosmosis, coupling of the proton motive force with generating ATP. That is what chemiosmosis is. So yes, both of them are using chemiosmosis. They use different energy sources. The plant uses light, solar energy, and mitochondria uses chemical energy because they're breaking down glucose to derive all of that energy. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into the chemical energy of ATP. Spatial organization of chemiosmosis differs between chloroplasts and mitochondria, but also shows similarities. That's just what I was showing you here. In mitochondria, protons are pumped to the intermembrane space to drive ATP synthesis, and then they diffuse back into the mitochondrial matrix, whereas in chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. ATP and NADPH are produced on the side facing the stroma where the Calvin cycle takes place. Yep, so Calvin cycle occurring over here in the stroma, again, very important. And in summary, light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. Okay, so finally, we're going to go over the Calvin cycle. So the Calvin cycle is where we're going to get our payoff. Remember, plants that have chloroplasts that go through photosynthesis, their job is to turn solar energy into chemical energy, right? So their job is to make molecules, organic molecules. To do that, they also have to create energy. So we just saw how they create energy. And now in the Calvin cycle, we're going to see how they take that energy and turn it into, well, turn carbon dioxide into sugars. So this is going to be the reduction. Carbon dioxide is going to be reduced, gaining electrons, and in the process is going to build a larger molecule, the sugars. The purpose of cycles in metabolism is to regenerate what it starts with. And the Calvin cycle is no exception. We saw a cycle in cellular respiration, which was the citric acid cycle, or the Krebs cycle, which started off with acetyl-CoA, and then we ended up with oxaloacetate, and it was a cycle we were creating citric acid. So the Calvin cycle is also a cycle. So it's going to be able to regenerate its starting material after all of the molecules and all the energy molecules enter and leave that cycle. Remember, we're building a molecule here. We're building sugars, and this is anabolism, and an anabolic pathway. That is a pathway that is building. Catabolic breaks down. That's cellular respiration. We were breaking down glucose. Here we are building up glucose. So this is an anabolic cycle. So we're building sugar from smaller molecules. We are using ATP and the reducing power of the electrons that are carried by our electron shuttle, which for photosynthesis is NADPH. We are going to go over 3 phases of the Calvin cycle. The first phase is the carbon fixation, and we're going to talk about an enzyme called Rubisco, which is an important one to note. Then we're going to go into the second phase, which is the reduction phase. And remember, reducing means gaining electrons. And then the third phase is the regenerating of CO2 acceptor, RUBP. So this is the part of the cycle where we are regenerating what we started with so that we can continue our cycle. Remember, we always want to track our reactants and our products. So for the Calvin cycle, what we're beginning with is carbon dioxide and ATP and NADPH. And what we are creating is a sugar called glyceraldehyde 3-phosphate, or G3P. You may recognize this. I called it out in the last lecture when we were going through glycolysis. If you look back, there's 10 steps in just the first part of cellular respiration called glycolysis. You'll notice this this molecule here, G3P. So it pops up in different metabolic pathways. So we are going to take carbon dioxide and reduce it to create a sugar called glyceraldehyde 3-phosphate, which is G3P. You also need to note how many turns of the cycle we need in order to create what we're trying to create. So for 1 G3P, the cycle has to turn 3 times, and we'll see that. So here's the basic cycle, the basic Calvin cycle here. You don't need to know every part of this. I'm going to call out the parts that you need to know for my class. So make sure that you note those, but you don't have to note everything that's on here. So first of all, let's take note to the 3 phases. Phase 1 is the carbon fixation. Phase 2 is reduction. And then phase 3 is regeneration. So what does it mean to fixate? You may hear that trees are our carbon fixators. What that means is trees and plants are able to take carbon dioxide, which obviously contains the atom carbon, and is able to pull it out of the air and fixate it, use it to build other molecules. That's the whole point of what we're talking about here, right? But that's really important. We always talk about the increase of carbon dioxide and the release of carbon dioxide there's a lot of carbon dioxide held in our oceans, and the heating of our oceans releases gases, particularly carbon dioxide. So trees are important because they can fixate that. They can snatch the carbon dioxide molecules and use them to rebuild materials again, chemical energy. So that is what carbon fixation is. So this first part, the plant is taking the gas, carbon dioxide. Remember, this diffuses through the part of the plant called the stomata. And so the carbon dioxide comes through, and the enzyme, there's a special enzyme called rubisco. Rubisco is short for ribulose bisphosphate carboxylase, so it's a big word, but we shorten it up to rubisco. Rubisco grabs a carbon dioxide and places it onto this molecule right here, which is ribulose bisphosphate, RUBP. So we always track the number of carbons. So this is, each one of these gray circles is a carbon. So ribulose is a 5 carbon sugar, ribulose bisphosphate. Bis means 2, like a bicycle, biphosphate. So there's 2 phosphate groups on this ribulose. So here's ribulose bisphosphate, 5 carbon sugar. The rubisco molecule is going to take one of these carbon dioxides, which just has 1 carbon, and add it onto here. So rubisco creates a 6 carbon molecule. See, 1, 2, 3, 4, 5, 6. Then another enzyme is going to cut that in half, right? So we have 2, 3 carbon molecules. You don't need to know the name of this. Just know this first step. Rubisco grabs a carbon dioxide and adds it to the molecule ribulose bisphosphate, which is a 5 carbon sugar, to create this 6 carbon sugar. That 6 carbon sugar gets chopped in half. And then we need to use a little bit of that energy that we made in the first step. So here's an ATP. We hydrolyze that ATP, ATP hydrolysis, right? We chop off one of those phosphates and we're going to add it on to this molecule. That charges it up so that we can, that gives it energy. Remember, breaking up ATP is an exergonic reaction. So you couple that with an endergonic reaction to drive this reaction. That's just what ATP does. So again, you don't have to know these steps. I'm just explaining it to you a little bit. That creates a new molecule called 1,3 bisphosphoglycerate. This molecule then gets reduced. Remember, reduction is gaining of electrons. So here's our NADPH that we had created in the light reactions. It comes on, drops off its electrons. And there's a couple of steps that happen here. And what it does is create this sugar, glyceraldehyde 3-phosphate, the one that I talked about in the previous slide right here. So we began with carbon dioxide. Rubisco fixated it onto ribulose bisphosphate. We used some ATP. We used some NADPH. This is the reduction phase, by the way. As soon as NADPH comes in with the electrons and starts dropping them off, that's reduction. So here's the reduction phase. We did fixation, reduction. We created glyceraldehyde 3-phosphate, and this is our output. So G3P, glyceraldehyde 3-phosphate, is a 3-carbon sugar. We know that glucose is a 6-carbon sugar, right? C6H12O6, that's glucose. This is not glucose. This is just another sugar that's going to be used to build glucose and other sugars. Plants make more than just glucose, right? We know that they make cellulose and starch and other ones that I can't even name. So this is just a little building block that can be used to build all these other sugars. So this is the output G3P. Now, we need to regenerate this ribulose bisphosphate, otherwise this won't be able to continue that cycle. So well, let's back up a second. What's happening? In the previous slide, we said that to build one G3P, that cycle turns 3 times. So it's not that this particular one's going 3 times. There are 3 of these occurring simultaneously. So 3 carbon dioxides are being fixated onto 3 different ribulose bisphosphates. And to be honest, this is a very confusing cycle. If you want to see it, there's a really good video on it. If you want to see a really good quick video on this, if you're a visual learner, look up Nature's Smallest Factory, the Calvin cycle. It's a TED education video, and you'll notice this guy, this is Rubisco, that enzyme. It's taking carbon dioxide and it's about to add it onto this ribulose bisphosphate. But this video shows very well what I'm talking about, how we actually have a bunch of the cycles happening at the same time. So this would be 3 of them happening at the same time to make one glyceraldehyde 3-phosphate. And that's just a 3-carbon molecule. So if we want to make glucose a 6-carbon molecule, we need twice as many. So instead of 3, there are 6. And then this will make one glucose molecule. You don't have to know the details of that, but this is a great video to take a look at so you can see what I'm talking about. Essentially, to make one of these G3P, there are 3 of these cycles happening. If I want to make that glucose, I need 6 of these cycles happening. So what this does is it allows for the regeneration of RUBP. We're actually, if we do 3 of these turns, we're going to make one G3P, and we're going to be able to make 3 ribulose bisphosphates from it. So this is the regeneration of RUBP phase. Really, that's all you need to know. There's a little bit of energy used here as well. So the energy was used in the carbon fixation phase. The NADPH was used in the reduction phase. And then we have a little bit more energy used in the regeneration of RUBP, our 5-carbon sugar here. And that is the Calvin cycle. Just remember, Rubisco, no ribulose bisphosphate, 5 carbons. Know that we use energy and the NADPH. Know that you need 3 turns of the Calvin cycle to make one G3P, or 6 turns of the Calvin cycle to make 1 glucose. And note the products. So let's look back at this picture. Now when you look at this, it should look a little bit, everything should look a little bit more familiar and fall into place, right? The light reactions used light to energize electrons that then went down the electron transport chain. Through that, we were able to create ATP via chemiosmosis. The electrons got passed on to NADP+, which reduced it to NADPH. We split water in this phase at the first photosystem 2. Here we go. Water gets split at photosystem 2 to regenerate those electrons that we're sending through the electron transport chain. Note where everything's taking place. Once we split water, we release oxygen. Then our energy products here are shuttled over into the Calvin cycle, and this is all in the stroma. The Calvin cycle, we start off with carbon dioxide. Rubisco fixates that onto RUBP. That's the carbon fixation cycle. Then the NADPH comes in, reduces that. You know, there's a couple of steps as you saw. The end product is we create a G3P, glyceraldehyde 3-phosphate. That G3P then gets sent to other metabolic processes, pathways to create whatever sugars they need to create. 3 turns of this creates 1 G3P. 6 turns of this creates a glucose. And then we have the regeneration of RUBP step here so that we can continue this cycle. And what do we do? Once we hydrolyze ATP, obviously the products are the ADP and inorganic phosphate. And once we oxidize NADPH, we're going to take the electrons off of there. That means that we are generating just an NADP+, which is able to come back into the light reactions to gather more electrons. NADP and inorganic phosphates able to come back over here to be put back together to create more ATP. So this is a nice cycle here. Our input here also light, our outputs sugar and oxygen gas. So make sure that you understand that very well. You will see questions relating to this in different ways. So if you understand that, you'll be able to answer those questions. Okay, so that was your basic photosynthetic pathway with the light reactions into the Calvin cycle. But of course, there are many different plants that live in many different environments, and there are some alternative mechanisms that plants can utilize for energy or to, for their biosynthetic pathways. So we're just going to basically talk about a couple of things really quickly. First, I want to point out you might come across this term C3 plant, and then we have C4 plants. All that means is this is talking about that carbon fixation step, that, this whole part right here. This is talking about that output sugar that they're creating. Remember, glyceraldehyde 3-phosphate, G3P, this is a 1,2,3 carbon sugar. So their cycle is creating a 3-carbon sugar. So these would be C3 plants. And some plants, and this is most plants, and then some plants have a different mechanism and they create a 4-carbon output, and those would be C4 plants. So as you might imagine, there are many plants that live in climates that are very hot, arid, dry climates. In these cases, these plants need to conserve as much water as possible. So to do this, they're going to close off the pores called stomata. And let me bring up the picture here. These are the pores that the gases carbon dioxide and oxygen are able to diffuse in and out of. So the plants will actually close off the stomata which is able to conserve water. But this also is, as you can imagine, going to limit the amount of carbon dioxide that can come in. And we know that we need carbon dioxide in order to get the Calvin cycle going and create that sugar output. So in these situations, if they close their stomata, they're going to have a build-up of oxygen. This helps with preserving water as well, but it also limits the carbon dioxide that's able to funnel through. These conditions of high oxygen gas and low carbon dioxide gas favor a process called photorespiration that we're going to talk about really quickly. But just note with this that this is a trade-off that is occurring where they're able to be more hydrated, which is necessary, but they're not able to create their sugar output with this. So let's look at photorespiration. As we now know, the first step of the Calvin cycle for C3 plants, we are fixating carbon dioxide onto RUBP using the enzyme Rubisco to form a compound called 3-phosphoglycerate, which later is created into G3P. But in photorespiration, we have really low conditions of carbon dioxide, and instead we have a ton of oxygen. So oxygen is what gets fixated onto the molecule, and we don't create these molecules, we'll create different ones, and what that does is it creates a 2-carbon compound. But what this process of photorespiration does is it's able to consume all of that built up oxygen gas. It also consumes some of these organic fuels that we've created, ATP obviously, and ADPH, which is carrying our electrons. And it's releasing carbon dioxide, but it's not producing ATP from this process or sugars. There will eventually be the creation of G3P, but the efficiency is decreased by 25% when a plant is going through photorespiration. Now, because this process is using some of our energy, and it takes a lot to create this energy, and now we're utilizing it without creating a sugar, without creating more energy. So this process, photorespiration, is pretty wasteful, and they believe that it came about during a time when the atmosphere had far less oxygen gas and more carbon dioxide. So then photorespiration, when they close their stomata, they're able to conserve any oxygen gas that they did have, and they really didn't need that much more carbon dioxide for themselves. But what it does is it is able to limit the damaging products that the light reactions create, which could build up in the absence of the Calvin cycle. And in many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. Now, C4 plants are fixating carbon dioxide into a 4-carbon compound. Let's just look at the picture. So what they do, so this is the carbon dioxide that's coming into a cycle before it gets into the Calvin cycle. So it uses a different enzyme for this, PEP, phosphoenolpyruvate carboxylase. PEP has a much greater affinity for carbon dioxide than Rubisco does. So remember, Rubisco is grabbing the carbon dioxide in our regular Calvin cycle, but PEP carboxylase has a much greater affinity, so it's grabbing the carbon dioxide first, and it is fixating it onto a 3-carbon PEP, right? So we are creating a 4-carbon compound called oxaloacetate. Hopefully that word sounds familiar to you, because I told you need to know that one for the cellular respiration phase called the citric acid cycle. This is a molecule, an organic compound that's in that cycle. Actually, so is malate. And pyruvate is also present in at the end of glycolysis. So we have some common compounds here. So PEP carboxylase is grabbing carbon dioxide, fixating it onto PEP, and we create a 4-carbon compound. And then it is regenerated, the 3-carbon compound is regenerated, and this kicks out carbon dioxide into another part of the cell over here for the Calvin cycle to occur. These C4 pathways are special because they're able to minimize the cost of photorespiration by incorporating this carbon dioxide into a 4-carbon compound. C4 photosynthesis actually uses less water and resources than the C3 photosynthesis plants do. Scientists have actually been able to modify some rice, right? This is what GMOs are, genetically modified organisms. So they were able to modify a C3 plant into carrying out C4 photosynthesis. And they estimated that doing this increased yield by 30 to 50% in comparison to rice that is utilizing C3 photosynthesis. And finally, we have CAM plants. CAM is cressolation acid metabolism, CAM, and they use this to fix their carbon. So what CAM plants do is they open up their stomata only at night, and this allows the carbon dioxide to come in, and the CAM plants fixate the carbon dioxide into organic acids that they store in their vacuoles, and then during the day the stomata close down. Carbon dioxide is released from those organic acids and it's utilized in the Calvin cycle. So here's a little comparison between C4 and CAM. So notice how the first step, the carbon fixation, they're both taking the carbon and creating an organic acid. In the C4 plant, remember it was oxaloacetate, right? In the CAM plant, well, we don't really go into that here. So they create an organic acid, and then the carbon dioxide is again released in that cycle, and then it's fed into the Calvin cycle. This didn't happen in the C3 plants. Recall that carbon dioxide comes in right at the beginning of the Calvin cycle, and then that was it. So these are a little bit different. Couple things that are different here. These two pathways are very similar in that they both incorporate CO2 into organic intermediates before they enter that Calvin cycle. But the C4 pathway structurally separates the initial steps of carbon fixation from the Calvin cycle. These are in two different parts of the cell. This is in a mesophyll cell, and this is a bundle sheath cell, two different types of cells. Whereas this doesn't happen in the CAM plants, and in the CAM pathway, these steps are occurring in the same cell, but they're separated for time. So at night is when the stomata open and the carbon dioxide comes in and it's fixated into organic acids. And then during the day, the organic acids, the stomata are closed. The organic acids release carbon dioxide. It's not going to diffuse out because they're closed. It's going to be fixed with Rubisco in the Calvin cycle, and it's going to create sugars. So this has spatial separation of the steps, and CAM has temporal, meaning time, separation of the steps. And that's it for photosynthesis. I know it's always a lot when you first learn cellular respiration and photosynthesis. The best thing to do is to create tables, to log all the things that I pointed out to you. Always note the reactants, what we're beginning with, the products, know which carbon-based molecules are going into something and which are coming out, when and where are ATP created and used, when and where are NADPH created and used, where are the reactions occurring, right? This again is a really good snapshot of everything. Know when light comes into this. Light is not used in the Calvin cycle. We have two different photosystems for the light reactions. Make sure you know which one uses water, right? PS2. which of the chlorophyll A pigments are in each one. Photosystem 2 has P680, photosystem 1 has P700. Where is ATP made? So this is linear electron flow. Then we can have cyclical, where this cytochrome ferredoxin shuttles around and the electron is just shuttled in a cycle. And by doing that, we are creating extra ATP, but no NADPH. Details like that, that's how you're going to see these questions on an exam. Definitely know the similarities between the mitochondrion and the chloroplast, because they are so similar. Be able to explain this from start to finish. This is the light cycles, the light reactions. Note that ATP and NADPH are both created in the stroma, which is where the Calvin cycle is taking place. For the Calvin cycle, there are the 3 different phases. Note Rubisco, we're fixating carbon. Carbon is being reduced in the reduction phase. We're creating a G3P. We need 2 G3Ps to make a glucose. Each 3 turns of this creates 1 G3P. 6 turns creates 2 G3P, which we can create a glucose from. Then we have to go through the regeneration phase to create RUBP again, our 5-carbon organic molecule. And we talked about alternative methods of photosynthesis. We talked about photorespiration in sea plants when there's extra oxygen. So instead, Rubisco is fixating RUBP with oxygen, and then we're not creating this. We're creating a 2-carbon molecule. So it just reduces the production of G3P by up to 25%. It's very energy taxing. Then we talked about, and it's a protective mechanism for plants in dry, arid places. The whole point of it is to keep as much water as possible in the cell. That's why they close up their stomata. And we talked about C4 plants. C4 plants, their carbon fixation step, they're creating a 4-carbon compound. We talked about the CAM pathway, which again is also creating it is also fixating carbon dioxide into an organic acid. C4 and CAM are very similar, but they do have a couple differences. This is separated by space. They're in 2 different cells. This is separated by time. This occurs at night. This occurs in the daytime. And yeah, so go over that, and again, make sure that you also understand cellular respiration. Go over that lecture if you need to see it again, and I'll see you in the next chapter. Good luck.